Good morning.
Good morning.
How's everybody doing? Enjoy your 3-day weekend?
No.
No? No? Had a whole extra day to relax.
To work.
So we're going to start off, we're going to pick up where we left off all the way last Friday. We were looking at transcription factors and comparing, we ended up comparing enhancers and promoters, so we'll finish that up today and get into some RNA processing. Before we do that, some announcements. Just to remind you, excuse me. So exam 1, you have until a week from today for final point adjustment. So you have recitation this week to take the 2 particular questions, the one on the functional group. What was the second one? The one on the modified kinetochore motor protein. Take those to your PIs. If you have anything else you want to discuss as far as the exam or understanding why it was graded the way it was, I have office hours today, or feel free to email me and schedule another time. Okay, and I've taken care of, talked to a number of people, got some things cleared up. Please come in and talk to me if you're concerned about something. Check all your grades that are up so far. Make sure that your quiz and homework grades have been recorded properly. Make sure that your participation points are about where you think they should be. If you don't have participation points and you think that you should, please email me. You're probably one of the last few people who's got some registration issues. All right, taking care of a few of those, but please go in and check that. All right. So, oh, and just I think that I promised a number of you, the average on that first exam, as I currently have it calculated before all the adjustments and everything, is a 77%. All right, so pretty good. So that's exam 1. It's already time to start thinking about exam 2. So if you haven't been paying attention, your next exam is a week from Friday. Yes, it snuck up on us because of the 3-day weekend. All right, so start thinking about that next exam. I owe you a studying guide and a Mastering Biology, so I'll try to get that Mastering Biology up today. I'll get you the study guide by Friday, or sooner if possible. But you need to start thinking about what we've covered. We're going to finish up with transcription today, get into post-transcription processing, and start talking a little bit about amino acids, how you put them together, how you draw them, thinking about translation next week before the exam. All right, so start thinking about that on your homework. You had a warning, learn all your amino acids. I've posted on the Facebook page several YouTube videos designed to help you learn those, so take a look at those. If you come up with any better ones, please post them so that everybody can have that information. Start practicing now if you haven't already, okay? And we'll look at, probably on Friday, how to draw them, for this class anyway, and what I expect, so you'll have that going into the weekend. All right, so related to the second exam, the TIs will be having a review session again, just like last time, Tuesday night from 5:30 to 6:30. Hopefully in this room. I'll let you know once that's confirmed. All right, so please put that on your calendar. I think it was really useful for a number of you last time. It's a few days earlier than the exam this time. So you have some extra days to ask questions if you still don't understand anything. All right, that second exam will cover all the material through lecture on Wednesday. And finally, related to the fact that the second exam is on a Friday, there won't be any recitation that week. All right, so that gives you some extra time to study. And then you don't have to worry about it after the exam, going through a recitation when your brain is fried on molecular biology already. Okay? no recitation. Recitation for sure this week, if you want to go and get help from your TIs to prepare for the exam. No recitation next week. All right? Any time that your TIs make available for extra help is up to them. So if you ask them nicely, maybe they'll set up something else. But that's on their own time. Okay? But your TIs love you, so they might be willing to send you next time. Don't forget you have access to the Learning Assistance Center over in Sinclair. As well, you can get tutors through them if you need them. Okay? Are there any questions related to the exam? I'll post announcements once I start getting things posted on Mastering Biology and the study guide. Yes?
The 20 polypeptide structures, you want us to know them by exam 2, right?
I do.
So that's pretty much a week.
Yes.
For us to learn them. Okay, just making
Well,
sure.
I gave you the homework last week, so that gave you 2 weeks. You had 2 weeks notice. Yes. That's good.
You don't have to learn how to like decode them, like the specific sequence is.
So for this class, if I'm going to ask you to translate an mRNA, I will give you the translation table. You do not have to memorize that. You just have to know the translation. You have to know how to use the translation table. But if I give you a problem like that on the exam, I'll provide it for you. Okay? So know the structures, know how to use the translation table, but that will be provided. All right, any other questions? All right. So we'll see how far we get before the next exam, and I'll adjust the exam questions accordingly based on whether or not we get as far as I think we're going to. So I'm not going to ask you a question on material I haven't covered in lecture yet. All right. Any more questions before we start on transcription factors and finish that up today?
It's not cumulative, right?
Sorry?
It's not cumulative, right?
All of the exams, just to remind you, are cumulative. So it's fair game to ask you a question on any of the material we've already covered. Which means you can't forget how to draw your nucleotides, or any of your functional groups, or any of your bonds. Or the what?
Or the sizes.
Or the sizes, although we're going to stay focused on the structures. All right, the next exam. If I want you to know any numbers, I'll put it in the study guide. All right, so we may or may not start getting to proteins and interactions with some of those sizes, but we'll see how far we get. Any other questions? All right, so let's go back to, we were talking about the regulatory transcription factors on Friday. The difference between general transcription factors and regulatory transcription factors. So there's general transcription factors. Remember, there are a few that I specifically told you what their jobs were. Pay attention to those. They're important. For the regulatory transcription factors, so we're looking at proteins that bind to DNA control elements that are regulating the rate of transcription of a particular gene. Alright, so this is the schematic drawing of that. We have our general transcription factors, assembling at the promoter, recruiting the RNA polymerase, giving the go signal for transcription. And then we have all these other DNA elements that are binding regulatory transcription factors that are going to then fold back, interact with that transcription machinery, that initiation complex, and either enhance that rate of transcription or repress it. We were talking about on Friday what distinguishes an enhancer from a promoter. We got most of the way through that. So we looked at What happens, we ended with what happens if an enhancer is rotated. We looked at what transcription levels look like with a promoter, without a promoter, with an enhancer but no promoter, enhancer and promoter, enhancer close, enhancer far away, enhancer rotated, right? This is where we ended up. So just for comparison, because we're looking at an enhancer versus a promoter and how they function, let's look and compare what happens if you rotate a promoter, now that we've looked at what happens when you rotate an enhancer. So first of all, hopefully our clickers are working today. Based on this schematic, which is the template strand that transcribes?
Top. The top.
Is all the information I provided there? All right, everyone had a chance to answer? Look and see what the class thinks. Results are going to be hidden. All right, so we're going to put this. There we go. So we're looking at the direction of the promoter and the transcript that's being produced. Which strand has the opposite orientation? B. All right, so that's the correct answer. If the promoter is in this orientation, what if we rotate that promoter around? Now what's the template strand? Same one or the other one? Everyone get their answers in? Let's take a look. All right, so I think you guys have this concept figured out. All right, if you rotate the promoter, all right, in this case, the top strand is now the template. Means it's bringing in the RNA polymerase in the other orientation. It identifies the other strand as the template. You're transcribing something different. All right, so it's changing what's being transcribed if you rotate the promoter. All right, so the final question. Move the first thing up. What happens down here for G? We have our promoter. What happens if we put the enhancer on the other side of the gene? What happens to transcription levels? Think about how an enhancer is working. What you know about it already.
I wonder if there's a minus. Yeah, like it's so negative. You did it, that it's been already. Yeah, yeah.
Alright, everyone get their answers in?
Like your body would disintegrate.
Take a look. Alright, so a little bit of confusion. Does it matter where the enhancer is relative to the gene of the promoter? It doesn't. Remember, because it loops back in, it can be just about anywhere. Alright, so the correct answer is that transcription is still increased even when the enhancer is placed downstream of the promoter in the gene. Alright, so what do you know now about enhancers? So take a quick minute with the people around you. Make a list. Make a list of the features of an enhancer.
Enhancer non-directionality, no matter what they are. Bound by regulatory. No, but like.
Alright, everyone have their list? Give me a feature of an enhancer.
Non-directional.
Non-directional, is that what I heard?
No directionality.
Direction doesn't really matter. What else?
Placement doesn't matter.
Position doesn't matter?
Distance from the promoter doesn't matter.
Should nothing matter.
Anything else? Define what kind of transcription factors? Enhancers, specifically activators, but more generally, regulatory. Ready to read my shorthand?
Assigned to regulatory.
They're not necessary for transcription.
It's transcription, exactly.
But what do they do?
You shortened it. Anyone who consumes the
Not necessary for transcription, but they. What do they do then? Transcription levels? Increase. They interact with the general transcription. Well, transcription factors they bind interact with the general transcription factors to increase transcription levels. All right. So they're orientation independent. They act from a distance. Doesn't matter where they are positionally. Because that DNA bends to allow the interaction. They modulate the rate of RNA polymerase recruitment to bring in that RNA polymerase in, to help increase transcription. And they also modulate the rate of transcription initiation. So they're increasing transcription by helping to recruit polymerase, by helping to get initiation started. Okay? So how do enhancers differ from promoters? Two basic ways.
Enhancers are not required.
Orientation, distance. Promoters are required and enhancers are not. Orientation, distance, one is required, one is not. All right, so the last thing we're going to look at relative to transcription factors is So we've been talking about enhancers. Remember, enhancers are DNA elements, right? It's really easy to get the DNA element confused with the protein transcription factor. So we're going to look at the last thing is how transcriptional repressors, all right, that are binding to silencers. So the DNA element is a silencer, the repressor is a transcriptional factor, and how those can interact to help reduce levels of transcription, right? We're turning genes not only on, but also off. So there are two main ways that these repressors can interact and reduce transcription levels. One, it can block the enhancer site. So if we have our activator in red, our repressor in green, they have overlapping binding sites. There's DNA elements, enhancers, and silencers. So if we have a repressor bound, then the activator can't bind, which means it can't interact with the transcription machinery and increase polymerase recruitment or initiation. Right? So one way that repressors interact is just by outcompeting those activators for binding sites. At the level of the DNA recognition sequence. Another example of a way a repressor can help, well, can decrease transcription or keep those levels low, is that it can bind to its own site far away from an enhancer, but then it can bind to the general transcription factors and block activator interactions that way. So here we have a repressor in green. It's binding to that TF2D, which recognizes the Tata box and the promoter. Because it's bound there, it can inhibit the recruitment of polymerase and the rest of those general transcription factors, as well as the activators. All right, so depending on what's bound, it's going to either increase or decrease polymerase binding and transcription initiation. All right, so we've looked a lot at enhancers. We've looked a little bit at how there are also transcriptional factors that are repressors to give us this really fine scale control of gene expression and transcription. The final thing to look at for transcription factors, I think the final thing, is actually what these transcription factors look like. We know that they are proteins, that they are binding to recognition sequences or DNA elements. We know that they have two domains, a domain that recognizes that DNA element that it's going to bind to, and a domain that then interacts with other proteins to control transcription. And there are three general motifs that are common in these regulatory transcription factors: helix turn helix, zinc finger, and leucine zipper. So let's take a quick look at those three, just so you have an idea of how these transcriptional factors are interacting with the DNA. So to remind you from the last unit, we talked a lot about the major groove. Here's an example of how a protein is interacting in that major groove to recognize a particular base or a base pair. So we have our regulatory protein, we have asparagine, an amino acid. Here's its side chain, right? So we have a hydrogen donor and a hydrogen acceptor, and it can form hydrogen bonds at those functional groups. All right, so these transcriptional factors are in the major groove, binding with the proper base pairs. That means that they have to have a recognition sequence in the protein. So the helix turn helix motif, so we have two alpha helices with a turn in the middle, much like the name suggests. One of those is a recognition helix that is going to sit in that major groove and bind with a specific sequence of DNA. Right? And the residues and their side chains are going to form hydrogen bonds with the proper bases in that major groove. All right, so here's two examples of a recognition sequence And then that second helix, one of those sitting in the major groove, interacting with the beta. The second one, the zinc finger. All right, so the last one was 2 alpha helices. You had to think a little bit about protein structure for your homework. Here's an example of a transcriptional factor that's a beta sheet, anti-parallel in different directions, and an alpha helix. And they're held together in this finger form, this projection, by a zinc molecule that interacts with 2 cysteine and 2 histidine residues, the side chains of those. So the cool thing about the zinc finger that I think will really help display how proteins can find a particular recognition sequence in the DNA. So the side chains in this alpha helix are going to bind to specific amino acids. So it's only, or I'm sorry, specific nucleotides. So that's how you get each alpha helix recognizes 3. So by stacking up zinc finger domains with different amino acids with different side chains, they're going to bind to different combinations of nucleotides in that major groove. And so you can begin to build up a recognition sequence that way. And then finally, our leucine zipper motif is 2 alpha helices coiled together. There are leucines that interact along the top, and then they have these DNA recognition sequences like a tuning fork or a prong on the end that interact with that major groove. All right, so these are 3 examples of transcriptional factor structures, protein structures, and how they have that DNA recognition domain, and then another domain that is free to interact with other proteins and the general transcription machinery. All right, so are all genes expressed in all cells? No. There are lots of examples of this. Just looking at the globin, from fertilization through birth and after birth, we have all sorts of globins, some of which are expressed all the time, some of which are expressed only before birth, some only after birth. All right? Transcription factors control this. So if we look at 2 particular tissues, or why some neurons are expressing some genes and others aren't, we looked at the albumin gene. The difference between 2 different tissues, right, the albumin gene is produced by the liver. The difference isn't in any of the general transcription factors or the DNA sequence. It's in these regulatory transcription factors and which one of those are present in particular cell tissue types. All right, with those regulatory transcription factors in the liver, this gene is turned on and transcribed at high levels. In the brain, without those transcription factors specific to this gene, right, you get very low, just base level transcription. All right, so we've gotten through transcription and what's controlling transcription in various cell types and why they're different, why you can have 2 tissues that express some of the same genes and then also very different sets of genes, all controlled by these regulatory genes, regulatory transcription factors. So we've gotten through this DNA makes RNA step of the central dogma. We're going to move next to the mRNA makes protein. So we're working our way up to protein. But there's one step in between that we have to cover first. All right, we have to get from RNA to mRNA so that we can use the mRNA to make protein. So all we talked about now is general transcription, how the cell is making RNA. It doesn't matter what kind of RNA. We talked about all those different kinds of RNA. So for the rest of today and probably the beginning of Friday, we're going to talk about RNA processing. And this is extremely important, particularly in eukaryotes. All RNA is processed before it becomes mRNA. And in some eukaryotes, like the squid, they seem to be able to modify their RNA even more depending on changing environmental conditions. All right, so even though we have these things coded in our genome, once they're transcribed, lots of things can happen to them to affect what products are actually made. So another reason that squids are just really cool. You didn't know that already. So we're going to talk about post-transcriptional processing. So in prokaryotes, it's pretty straightforward. We have that gene that we put together. And in prokaryotes, the DNA transcribes directly to mRNA. The product of transcription can be used directly to make proteins. So that mRNA contains an open reading frame, or abbreviated as ORF. What do I mean by open reading frame? How would you define that? Any ideas? What's an open reading frame relative to, we're trying to make a protein.
Tells you the codons that code for the amino acids.
So it has the codons that code for amino acids.
Between the start and stop codons?
It's between the start and stop codons. All right, so the open reading frame is the part of that mRNA that is translated. Open reading frame means there's not any stop codons in the middle of it. So it's a stretch of nucleotides that code for amino acids, suggesting that there's part of that mRNA transcript that does not code for mRNA, or for amino acids. All right, and transcription in a eukaryote is a little more complex. What's transcribed is not in mRNA yet. It has to be processed before we can get to that step where we have an mRNA or some portion of that is an open reading frame that is then translated. So what happens to the transcripts in eukaryotes after transcription? There's a bunch of these processing steps that have to happen. And because eukaryotes have a nucleus, different stages in our central dogma process happen in different places in the cell. So we've been in the nucleus so far for transcription. The pre-mRNA, or primary transcript, is processed in the nucleus and then sent out to the cytoplasm for translation. And one of the major differences in prokaryotes and eukaryotes, prokaryotes don't have a nucleus. As I've already said, when a gene is transcribed, it's transcribed directly as an mRNA. So as an mRNA is being made, prokaryotic ribosomes can hook onto that and start making a protein immediately before the mRNA is even completed. All right, so you can have this mRNA being made from a gene, and you can have multiple ribosomes coming along behind the transcription machinery on that mRNA and making multiple proteins. So it's a fast process. In eukaryotes, right, we make our RNA in the nucleus. We have a bunch of processing we have to do in the nucleus. Then it has to get sent out to the cytoplasm, and then it gets translated. All right, so in the prokaryotes, it happens all at the same time. In eukaryotes, it happens at different times in different places in the cell. So what we actually transcribe in eukaryotes is a primary transcript, or a pre-mRNA. Hopefully, as you found out for homework, we have to do 3 things to that pre-mRNA before we can call it an mRNA and send it out into the cytoplasm to be translated. We have to put a cap on it. We have to splice it together to get an open reading frame, cut out the bits that aren't coding sequence, and we have to polyadenylate the 3' tail, put a poly A tail on. So if we think about that primary transcript, what it really looks like is this. So we have some bits that are going to be part of our final mRNA, and some bits in black that we're going to cut out during processing. So our primary transcript, or pre-mRNA. All right, we have exon-intron structure. Exons being the parts that are kept and spliced together. Introns being the parts that are cut out and removed. Seems a little bit opposite of how they should be named, right? You would think you would want to keep the introns because they're in, but it works the opposite way. And through this processing, what we end up with is an mRNA that has a 5' cap, that poly A tail, and an open reading frame. Again, just like in the prokaryotes, only part of that mRNA is translated. Right? So we have coding sequence and non-coding sequence in every mRNA. All right, so we're going to look at capping, we're going to look at splicing, we're going to look at polyadenylation, and how all of these things happen. mRNAs are not fully mature, and they don't leave the nucleus until all three of these things have happened. All right, the splicing gets the open reading frame together. The capping helps protect that mRNA from degradation once it gets out into the cell, where there are lots of enzymes whose sole job are to degrade RNA. As does the poly A tail. All right, so we'll get through capping, and we'll start a little bit on splicing, and then we'll pick up with splicing on Friday. So capping, you had to draw your 5' cap for homework. So you need to be able to draw the structure for the exam. All right, this is building on what we did in the last unit. Some things I want to point out so that everyone is aware of the important structural components of the cap. So first of all, you're going to see lots of different drawings of this and different orientations. The things that I'm going to be looking for. So first of all, notice that it's a 7-methylguanosine. Right? So it's a guanosine with a methyl group added on. So you've already learned what guanosine looks like, what guanine the base looks like. So you need to know where to stick that methyl group. The next thing to notice, back, question?
Between the two pictures, the O minus, there's no double bond, but in your second picture there was a double bond.
Say that again?
Okay, so the picture for this one, the 5' cap, the 7-methylguanosine, there's,
Oh, the O minus?
yeah.
I would take an O double bond.
Oh,
Yes.
so there's no preference?
I think, so for you guys, you know guanosine, draw guanosine the way you know it and stick a methyl on it. Yeah? Okay? All right, so methyl on the guanosine. Also notice, so here's one. There you go, with the double bond for you. So I'm going to show you a couple of different ways that it's drawn in class, and hopefully you will recognize the important components of what I'm looking for. All right, so here's another way to draw it. Also notice that in the cap, the first two bases, that 2' hydroxyl is methylated. All right, question? You do. Yes. So this whole process is to help protect the mRNA from being degraded. So that gets rid of a functional group, right? Protects those open hydroxyls. All right, so we have methyl now in the guanosine. We have methyls on the 2' of the first 2' hydroxyl of the first two bases. And then the other really big important feature here to notice that it's a triphosphate linkage, 5' to 5'. All right, everyone see that? So we have our 5' carbon on our first base. Notice that the linkage here is that this ribose is flipped. All right, this nucleoside is flipped around, nucleotide. All right, so that 5' to 5' linkage is why you see all these different orientations when you see drawings of it, because they're trying to represent that linkage and the orientation of the ribose and the base relative to the rest of the nucleic acid strand. So we have methylated guanosine, methylated 2' hydroxyls on the first two bases, and a triphosphate 5' to 5' linkage for our cap. All right, that's what we're going to be looking for when you draw, if I ask you to draw a 5' cap. Okay? Question? This bond here? Right, so in this bond, I accept either here or here, either way. It's essentially the same thing, drawn slightly differently. Well, I accept either. All right, one more look at a cap. So we've been looking at the triphosphate bond drawn horizontally. Here's a version of it drawn vertically, if that's your preferred way to draw your triphosphate. Notice it has the same features, methylated. Got an extra hydrogen. I don't know what that's doing there, ignore that. We've got an extra, we've got a methylated guanosine, methylated first two bases, 5' to 5' linkage. All right. All right, so I think we will end there. We'll pick up on Friday with splicing. Drink lots of coffee because splicing is complicated and often very tricky to keep track of what's happening on. So come to class Friday awake, ready to think about splicing, and have a good day.